What is an antibody and what is its structure? If you want to know all these things, then this video is for you. Watch the video till the end. Hello and welcome to a new video of AVIJIT'S Biology. And in this video I am going to discuss about the antibody and the structure of antibody. First of all, we have to know what is an antibody. So antibody basically is a protein, and it is generated when any unwanted substances is introduced into our body. When any unwanted substances, or it is known as the antigen, or any pathogen is introduced into our body, then antibody is generated to give protection against it, as it kills, or the antibody kills the antigen or pathogen. This is how antibody plays an important role in immunity. Antibody is also known as immunoglobulin or Ig. And now we are coming to the structure of antibody. So antibody is a Y-shaped protein. Basically, these antibodies are found in blood. Now let's discuss about the structure of antibody. An antibody looks like this, an Y-shaped structure. And this antibody consists of two regions. One is variable region and one is constant region. The upper part of the red line is the variable region and the lower part of the red line is the constant region. The name variable region suggests that the structure of this area varies with the shape of antigen. The structure of this region varies with the shape of the antigen, and the shape of constant region remains constant all times. So this is known as the constant region. And there are two pairs of chains in an antibody structure. One is heavy chain and one is light chain. Large two chains are the heavy chains and the small two chains are the light chains. Now this portion is known as the antigen binding site, as here the antigen binds, and the structure of the antigen is complementary to the structure of the antibody's antigen binding site, and this antigen binding site is also known as the paratope, and this paratope binds with the epitope of the antigen. Now this part of the variable region is known as the VH, that is the variable region of the heavy chain, and this portion is known as VL, as this is the light chain's variable region. Now the constant region of the light chain is known as CL, but the heavy chain have three different constant regions. One is CH1, another is CH2, and the third one is the CH3. But there is an exception. The IgM and IgE consist of CH1, CH2, CH3 and CH4 also. But IgA, IgG and IgD has three portions of heavy chain, that is CH1, CH2 and CH3. Now there are disulfide linkages or disulfide bonds between one heavy chain and one light chain, and between two heavy chains also. Now there are interdisulfide bonds between VH, VL, CL and CH1, CH2 and CH3 also. Now the region between CH1 and CH2 is known as the hinge region. This region gives the flexibility to antibody to bind with an antigen. IgM and IgE lacks this hinge region, so that they have another extra CH chain, that is known as CH4. As the antigen binding site is shown here, the antigen will be looks like this, which can attach to this site of antigen binding. And see that the antigen has a similar structure as the antigen binding site. That is how antigen-antibody reaction occurs. So far I have discussed about antibody and the structure of antibody. So that's all for the video. Please like the video and share the video, and please subscribe my channel for such videos. So we'll meet you in the next video. Thank you.